Hey, everybody. Okay, today we are going to be talking about wetland biogeochemistry. And this is really important. If you're going to be doing anything in wetland environments or actually as a marine biologist in any coastal environment, this biogeochemistry is extremely important because it is so important in terms of feeding that environment. That's going to be funny later, so remember that. Okay, so we are still in the part of the course that is in that coastal environment section. Today, we're going to be talking about wetland soils. Next class, we'll be talking about nutrient cycles. And what want you to get out of these combination of lectures is essentially you should be able to identify key characteristics of wetland soils and describe how to measure them. And you should also be able to describe global nutrient cycles and the role that wetlands play in the key nutrient cycles. Okay, so let's work through a couple of definitions. Soils. These are bodies comprised of a mixture of mineral and organic solid matter. They can also have liquids in them and gases, and all of this occurs on the land surface. So this is the exposed land on top of the continents. So you can think dry land there. And this is material that's derived in place from parent material that might come from the surface. That tends to be more organic material, or it might come from inorganic sources, which oftentimes is the parent rock in the lower levels of soils. And we're going to look at the layers of soils here in a little bit. In fact, you can actually see some layering in these soils, right, where you've got different matrices. And if you look up towards the top, you can see how dark and rich that is. This tends to have more organic material, living organisms in there, for instance, as well. You can see roots protruding down into the soils, where down here you see more rock-like material. Sediments are essentially soils that are underwater. Oftentimes, these soils have been transported and moved by either fluids. So, for instance, river flow. This is the Brazos River. And you can see all of the material that's being moved. The water is brown and murky, so there's a lot of material up in the water column. You can also see accumulation in some areas. West Texas would be a good example of this. It can also be moved by wind. And actually, here in East Texas, wind-driven movement of sediment or soils rather helps those soils become part of our overall soil matrix. So I don't know if you've ever noticed some of those hazy days we get in the summer. Oftentimes, our wind-driven dust all the way from the African continent that moves over the Atlantic Ocean and eventually deposits here in Southeast Texas. So the soils, especially very light particles that get up in the air, can stay there for quite some period of time, move long distances. So the difference here really, again, is sediment really is soil that is underwater. In many instances, this is moved. In some cases, this can be in situ, and we'll talk a little bit about that. All right, so we've talked about wetlands before. These are transitional areas between terrestrial and aquatic systems. These are inundated or saturated usually by surface water, sometimes by groundwater, depending where you are, some combination of those two. But it has to happen for a duration and or at a frequency that allows those soils to become hydric. And we've talked about hydric soils before. So hydric soils support vegetation that is specifically adapted to those environments. Hydric soils often are more anoxic, like we've talked about before, and the vegetation that grows there has to be able to survive under those types of saturated conditions. All right, so when we look at the features of these soils, one of the things we look at is the organic content, how much organic content is there. Organic content is derived from everything from plant matter to soil microorganisms. and the matter that those microorganisms are decomposing as part of their work in those soils. The microorganisms themselves become part of the soils as they go through their life cycles as well. The more organic the material, typically the lower density the soils are. So this density is really important. We measure this in usually a unit of mass or weight over a unit of area. So grams per centimeter per cubic centimeter, for instance, is one example of those. Um, and really what this is an indicator of compaction. So if you look here off to the right, you can see these soil particles, or they're supposed to represent soil particles, are not very compacted. You can see a lot of space between these particles. So they have a lower bulk density. So the grams per cubic centimeter of soil would be less, right? And these spaces, referred to as interstitial spaces, we've talked about that before in class, are really important in terms of how water flows through those soils. The larger the interstitial spaces, the easier it is for water to penetrate down into those soils, and they're often referred to as high permeability. Okay, this is important for a variety of things, including how nutrients move through those soils. On the other hand, if you look at compacted soils, they're referred to often as high bulk density soils, and they have smaller pore spaces, which means they typically have lower permeability. It's harder for water to get through those soils. So a lot of our soils here in Southeast Texas are low permeability, higher bulk density soils, composed of a lot of clays, Beaumont clays, for instance, that compact quite a bit. Um, Organic soils typically have an organic content that is 20 to 35% or above, okay? Whereas mineral soils that have lower organic content and higher bulk density tend to be at less than that, right? Um, so we can measure the organic content of soils. And one way of doing this is loss on ignition. So organic material vaporizes at approximately 400 degrees Celsius. So if you weigh a sample, a dry sample, by the way, so if you dry a sample in an oven, say at 100 degrees Celsius for 24 hours, and then you weigh it, then you expose it to a 400-degree oven and then weigh it again after that exposure. The difference is reported in percentages, and that represents essentially the amount of the organic material that was volatilized by that high heat. Okay, so I mentioned earlier layers, soil layers. And so soils are often easily, you can see in a core, for instance, you can see different layers of soils. Now, I say this isn't always as distinct as I'm going to paint it in this picture because, again, this is a textbook picture. Sometimes you need it takes a little bit to differentiate, for instance, the O from the A horizon or the B from the C horizon. So as you move down from the top, these soils have less oxygen in them and less living material, right? So the O horizon here, which is usually the top 2 centimeters I'm sorry, 2 inches or so, these are typically going to have the most living organisms in them because there's the most oxygen there. You're going to have the highest level of organic content in there as well. This is, the top of this often has what's referred to as a litter layer. So that's where dead leaves, decomposing plants and that kind of stuff on the surface that are just covering the surface are going to be found. So as that decomposes and works its way into that soil, there's a high degree of oxygen, high degree of soil microbes, other organisms. So these can be everything from little invertebrates, even sometimes to vertebrate organisms that are burrowing down into those soils. The A horizon is the next one. This can extend down typically around 10 centimeters. Again, understand this can really vary depending on where you are. This is the top mineral horizon. So this is where you're going to see some of the mixing that occurs from the parent material and the lower layers. This is important because a lot of times this is where some of the most valuable nutrients are in the soils, especially things like phosphorus, which a lot of times are derived from that rock underneath the soils. Then you've got the subsoil horizon, and then you get down to the parent material, which again, oftentimes is a lot of rock. Now, that organic layer, if it is greater than 40 centimeters, right? So if you think about 10 centimeters being 4 inches, right? Um, and get kind of a sense of what I'm talking about. If it's greater than 40 centimeters, we refer to this as histosol. And this is formed when decomposition and mineralization of organic material is slowed down because of the organic conditions that are present. So think about hydric soils that we've talked about previously. And so these are oftentimes occurring in cool or even cold humid conditions also. Um, so otherwise, it's going to be referred to as a mineral soil, okay, with an organic layer on the top of it, like we saw over here, this O layer. Now, again, like I said, this is pretty distinct when you look here where you've had these mineral deposits here actually suggest that this was covered in water at one time. But you can see distinct layers as you kind of work your way down. But even here, it's difficult because of the arid nature of this area to discern the O horizon by just looking at this eroded side of a cliff. I mean, you've got to look at them up close. There's some challenge to identifying these. So we can identify wetland soil features based on their grain size. And so this refers to the sand, silt, and clay components in those soils. Now, there is a fourth component, gravel, which is larger than sand. You don't typically see it incorporated in here because it's not as critical in terms of how plants utilize these soils. For instance, what you're seeing off here to the right is a soil texture triangle that was developed by the U.S. Department of Agriculture to help farmers determine what kind of crops would grow best on their soils. Now it can be used, for instance, to determine how you might amend those soils even to get the type of soil you want in order to grow the type of crop you want, right? Again, because the soil texture, the size of that grain, how it compacts is really important to how water moves through it, how it retains nutrients, those kind of things. Okay, so for instance, if we were using this, if we had a soil here, if you went along this bottom axis and we had a soil that we measured to be 60% sand, 40% clay, which is up here, and 10% silt, then if I followed this up 60, right? And so that's here, and we've got, let's go down here to 10%. Um, Here we've got 10% silt. So we're right here in the sandy clay loam, right? Um, which, these descriptions again are meant to kind of describe kind of a range of different soils that have characteristics that would be beneficial to certain types of crop species. Um, how we measure the grain size is largely through, well, there's a lot of different ways to do it, actually. In my practice, I mostly use the sieve method, where you can decant it essentially through these different sieves. So these sieves have different screen sizes, right, that allow different material. In fact, I happen to have here some sieve set, right? This is get it in the picture. There we go. This is a sieve set, and the sieves, right, have different screen sizes in them as you go further down. You go further down, the screen sizes get smaller and smaller. So you can see here, and by the time you get to the bottom, the lowest screen in here, it's really fine. Okay. So it's essentially capturing only the largest particles at the top, and it captures smaller and smaller particles as you go down. And then it catches whatever slides through all the way to the bottom in the solid pan, which is typically going to be silts and some clay in the very bottom of that. So that seed method utilized most often in a laboratory environment. We're utilizing a shaker of some sorts. There's a lot of varieties of these. You can do this by hand also. The soils need to be dried in order to work them through the sieves effectively. So you'll dry the soils. Usually that takes 24 hours at about 100 degrees Celsius. And then you put the sieves, stack them up, you put them on the shaker. It sits there and shakes them, and those kind of filter through there. And whatever your specified time is, then you can then go through and weigh the soil that's retained at each screen in order to determine how much of this is sand, how much of it is silt, based on the particle size, right? So some of these you might get, these top three or four might actually be classified as sand depending on the screen size, the opening in the screen. And then might have one that's silt or sand. You can size those based on the target soils that you're looking at. Settlement is another one. This works on the principle of density. I don't use this one very much, but you'll see this a lot of times in agricultural labs. There's another way to do this by feel, and some of the really old wetland biologists who practiced used to use this method a lot of times in the field. Wetland scientist, so I got my wetland delineation certificate from many years ago. He showed us how to use this method, and he used it quite frequently. He was actually very accurate with it. It is, you have to practice it quite a bit in order to get good at it. And essentially, you can determine it by working it through your fingers, determining a sense of the feel of the soil. So if it's got a lot of silt or clay in it, it might feel really silky and smooth. Also, how long of a ribbon you can push out, so you can actually develop a ribbon that looks like a piece of bacon that you're pushing out there, and um, how how much integrity is there as that ribbon gets longer can tell you about the content of the soils. That is not my preferred method, but there are wetland scientists and soil scientists out there that have been doing this so long that they're able to do that very effectively. I am not one of those people. I like the seed method. Okay. So this graph actually talks a little bit about that ribbon method, how you would go through there and actually do that by feel. Again, not my preferred method. But if you ever want to have some fun when you're out in a wetland, you can take a little bit of soil, follow this chart, and see how you do. I've worked with this with students in the past, but again, this takes some practice to get to. All right. So when we talk about soil features, salinity is, especially in these coastal wetlands, is very important. Now, I want to be clear that when we talk about soil or sediment salinity, it is not going to be the same as water salinity. Water salinity can fluctuate, like everything in the water, can fluctuate greatly depending on what the conditions are. So you have a rain event, you've got tides coming in, you've got different temperatures. All of these things affect the different abiotic factors of the water, and salinity is, in fact, one of those things. Evaporation, evapotranspiration can also affect and concentrate soils at or near the sediment surface. So that's something to consider. So we typically use pore water salinity, which represents the average conditions in those sediments. And so you can see that in this graphic here, where the water salinity can vary dramatically over a tidal cycle, for instance. So if this were, let me think about a creek, so Clear Creek, for instance, up here in the Clear Lake area. It can vary, fairly significantly, but the pore water salinity is going to stay the same. And this extends to a lot of things about sediments. Sediments tend to be far less fluctuant in terms of their characteristics, both abiotic and biotic, over a longer period of time. That's, for instance, one of the reasons I look at benthic macroinvertebrates quite a bit in my own research, because they are sessile and they tend to reflect the average conditions of the sediments that they live in over a long period of time. They're not moving around. They're not transitory. Like I said, they're sessile. And the sediment conditions also are not very transitory. So if you've had an organic contaminant release, for instance. That tends to stay in those soils for a long time and affect them for long periods of time, where it might be gone in the water column within a matter of hours or days, right? And that's reflected in those abiotic characteristics and also in the living organisms that can be found in those. Okay, so this is true of salinity as well. When we're measuring salinity in soils, you can use things like pour water sippers to get down there and get samples. Rehydration is another method, and then so you rehydrate the soils, and then you can actually measure the salinity of the water. So you're kind of doing an extraction in order to uh, get that salinity. And so you could use something like a refractometer or a salinity meter or what you like. All right, so let's dive into hydric soils here a little bit more. So hydric soils, and again, we've discussed them a little bit in class. So think back on some of the wetland discussions we've had, but they're formed under conditions where the saturation is long enough, typically longer than 7 days during a growing season. Okay, so that means we're not talking about being saturated so much in the cold part of the winter, especially in areas north of here where they might freeze. This is going to happen during the growing season when the plants are growing. And that's important because if it's saturated, remember, that's going to prevent oxygen from getting down into those soils, and it's more likely to cause those anoxic conditions. So the length of the inundation affects how anoxic those conditions are, and that can slow decomposition, allow organic material to build up, and that then can build up organic soils. Now, again, those anoxic conditions are because of low diffusion of oxygen into the soil pores. So you've got stagnant water, right? Um, and also, you're going to have respiration occurring. Remember, there are microbes down in those soils, and all living organisms need to respire. So, when you have that organic material, that tends to chew up, as they're going through their process of respiration, it tends to chew up and lower the amount of um, uh, oxygen that's down in those soils. Where you have wet and dry cycles, then that tends to lead to more mineral types of soils. And if you look at this here, this graph off to the right, or this graphic rather, um, the left-hand portion of this where my laser pointer is, this would be a hydric soil. This is a soil that has been anoxic. You can see the black characterization of the soil here, indicating that there have been sulfides in those soils. Here, you see a soil that's better aerated. Doesn't mean it hasn't been wet, but it's better aerated. You can even see what looks like rust deposits in here. The color of the soils can tell you a lot about the redox potential of those soils and some about how long they've been saturated. And here again, you can see on these sharpshooter shovels here, different soil layers and different types of soils that range from upland soils that are essentially dry all the way to very anoxic soils. Now, there are a lot of indicators of hydric soils. Um, Color is a big one. In class, I brought a Munsell soil book, which I don't think I have here in my office. I think my copy. Oh, yeah, I do. I have a couple different copies of this. So one down in the lab, I have one at home. Here we go. But I've got this one. So this is a Munsell soil book, and this soil book, it can tell you a lot. The soil color can tell you a lot about the conditions of the soil. This is a very traditional method. These are very expensive books, by the way. The pages can get wet, so you can put the soil right behind the page. It's like almost like writing the rain paper, only heavier. So you put the soil in the hole behind here, and you can compare the color, and that can give you a good sense of the redox conditions, saturation levels over time, and that. So if you ever take a delineation course, for instance, a good delineation course is going to cover how to use this, because it's a fairly common means of looking at soil condition over time. But again, remember, you'll see everything from black to gray soils that might have red streaks through them. So if we go back a slide here, you can see here this soil has been saturated at some point. It's wet, and there are actually iron deposits in here. So that's what that red is. A lot of times those are associated with where roots from plants have been at one point, but allowing oxygen to get down in there to oxidize that iron that's in those soils. And again, we've talked about the sulfur smell that you can get with anoxic soils. And that's that hydrogen sulfide coming off of there, kind of smells like rotten eggs. Okay, so a few minutes ago, I utilized the term histosol. So can you tell me if this is a histosol or no? Or do you even have enough information? Well, what would be that characteristic? Remember, it needs at least 40 centimeters of this hydric soil in order to be a histosol. Well, do you know what this is? Well, it might depend. If you can read this measuring stick, you could maybe tell that that would be a histosol, right? If you couldn't measure it, you might not have enough information here to make that determination. Okay, well, let's talk a little bit today now about nutrient cycles in these soils. Nutrients are chemicals or substances that an organism needs to live. And that can be any number of things. If you take a multivitamin, you're taking nutrients, right? Your food is full of nutrients. These are chemicals or compounds, again, that support generally processes in your system, right? So you need these things in order for metabolic processes to occur, for instance. And this is true of all living organisms. Um, they must be taken in from the environment. So even though you might be able to synthesize some of these things, you probably aren't going to be able to synthesize enough of them to support yourself. So that's why it's so important, for instance, for us to have a varied diet, right? So that you're consuming things that are going to support the intake of those nutrients that are going to support those biologic processes. Um, so typically the ones you see referred to the most are, and because these are the ones that tend to have the most outsized roles, are everything from carbon, nitrogen is a really big one, phosphorus is a really big one, oxygen. Um, and they're often found in compounds that include carbohydrates and fats. So whether you're, speaking as a human being, whether you're vegetarian or not, whether you're vegan or not, if you're consuming this range of different organic materials in your diet, if it's carbohydrate rich, if it has fats in it of some sort, right? Some sort of oils, that is going to provide you with those nutrients that you need to support those metabolic functions. So a lot of times you'll hear ecologists when looking at say a wetland environment, if we're lacking living organisms, especially plants, it's really obvious with plants. Oftentimes you'll hear us refer to limiting nutrients, right? A limiting nutrient is the nutrient that's in the shortest supply relative to all of the nutrients that that organism needs, right? So it tends to get used up first. In other words, the supply runs out before all of the other nutrients are exhausted. That doesn't necessarily mean it's the rarest. It means that it might be the one that's utilized in the greatest volume or amount by the residents of that particular biologic community. But what it does when it runs out is it limits the organism's growth or other metabolic functions. So this varies depending on what species you're talking about, depending on the habitat. But in marine habitats, and actually in many habitats, even in terrestrial environments, nitrogen and phosphorus, and I would say more often phosphorus, are the most typically limiting nutrient. So where do we get these, right? Well, they can come from a variety of sources that are natural sources and those that are anthropogenic. I've used this term before, anthropogenic, from human sources. So decay of organic material is a really important one in these wetland environments. As plants go through their life cycle, when they die off, they go into senescence. So Spartina alterniflora that you've seen in your labs out in the field, right? When it in winter, when it goes into senescence, that material dies off as it is broken down into the sediments. That material actually includes a lot of nutrients, including nitrogen and phosphorus, that then can be taken up by the plants again as they go through their next life cycle, or other species of organisms, right? Leaching from soil and rocks. And I talked about this earlier. Phosphorus is found in a lot of rock material. And so phosphorus is really important to plants as a nutrient. But the places you can derive it from are relatively limited. So leaching is a really important source of phosphorus in a lot of habitats. There are anthropogenic sources as well. There are a lot of artificial fertilizers that are um, manufactured and then put on crops. These things tend to be water soluble. So both nitrogen and phosphorus, for instance, are water soluble. So they can leach out of the soils and follow water flow out into wetland and other aquatic environments. Sewage is one. Combustion of fossil fuels, right? So when you combust fossil fuels, a lot of those nutrients, nitrogen especially, from the fuels themselves, get up into or actually the process of combusting the combusting the fuel sources. Oxygen combines with nitrogen in the air, and you get things like nitrogen dioxide, for instance. Those eventually settle out into water or soils, and then that nitrogen can be separated by bacteria and become a nutrient. So a lot of sources, you want enough, you don't want too many, right? So another clicker question. Which of the following is a correct statement about limiting nutrients? Well, if you look at this, you would see that B, nitrogen, is a common limiting nutrient in marine habitats. Limiting nutrient is not necessarily the absolute lowest abundance. Sometimes it has more to do with how much of a given nutrient the community is using. Most plants use more nitrogen than phosphorus. Nonetheless, phosphorus sometimes and often is the limiting nutrient, but it isn't always the limiting nutrient, depending on the species of plants. The limiting nutrient is used up last relative to the other essential nutrients. Well, that's obviously not true because it's the first one that's used up. Okay. So another so I've been talking about nitrogen and phosphorus, but carbon is also an important nutrient. It is the basic element of all life forms. If in your chemistry classes you've heard about organic compounds, these are not well, they could be, but they're not necessarily the things you find in the organic section of the grocery store. Uh, when we talk about organic in a chemical sense, it contains carbon, right? And carbon atoms readily form fairly strong bonds with other carbon atoms. And they create these long, complex molecules that include atoms of other elements, right? So, and they build things like proteins, like carbohydrates, fats, nucleic acids, right? That are part of your DNA, your RNA. Um, they're an abundant nutrient, and typically they're not a nutrient that's going to be limiting. So we talked a little bit about organic carbon, right? And so when we talk about organic carbon, this is carbon that was once in living matter, okay, contained in a living organism. And you can usually recognize organic carbon because it's connected to hydrogen in some form or fashion, right? So If you look at this graphic here, so this is a graphic of methane, and methane CH4 is one of the most basic hydrocarbons among all of the hydrocarbons. So when you look at the carbon chains that make up things like gasoline or oil, they're long chains that include a lot of carbon, hydrogen, hydrogen connected together. So a lot of methanes that are kind of connected together, if you will. These are derived again from the tissues of once living organisms. Um, so I like to joke when I'm filling up my motorcycles, I'm getting a fresh batch of dinosaurs. In fact, it's just mostly a uh, fresh batch of old plant material that's decomposed. There are microbes that can be part of that as well, but it's again living organisms that then are broken down into sugars, lipids, amino acids, and the like. Inorganic carbon, on the other hand, does not directly come from living matter. So a good example of this is carbon dioxide that you see over here, right? Um, Again, carbon is the central feature here, but inorganic carbon, again, typically does not have that hydrogen associated with it. And this is part of the carbon cycle. And the carbon cycle includes more than just living organisms. Rock can have carbon in it, right? Limestone is a great source of carbon into soils. So there is a carbon cycle. And so when we talk about, you should have actually, as I'm sitting here thinking about it, you should have looked at nutrient cycles in your freshman, sophomore level biology courses. So biology 1406, 1407. Um, you might have looked at it in other courses as well. But when we talk about the carbon cycle, um, generally cycles indicate that there is, these things are moving around. The law of conservation of matter. Matter cannot be created or destroyed. So you've got this material that's moving around, and it moves from the atmosphere into the soils or into the water, and then from the water or soils back into the atmosphere, and you got this kind of continuous thing, right? And so you see this in the oceans where carbon is moved in and out of the water through a whole variety of processes. There's similar processes at the terrestrial level. Again, I'm not going to go through all those again. It's something you should probably have in your background. Go back and take a look at this if you need to. But one of the sources, the anthropogenic sources that has tipped the balance of the really tip the balance of the carbon cycle is our utilization of fossil fuels. A lot of the carbon cycle, both in the aquatic and the terrestrial phases, involves plants that sequester carbon out of the atmosphere, right? So they take carbon out of the air and they incorporate it into their biomass, right? Because they're using that carbon to build sugars with other nutrients that they're pulling in through their roots. All right. So they pull in carbon dioxide, they strip off the carbon, and then they exhale oxygen, O2, back out into the atmosphere. All right. And this occurs in the ocean as well to an even greater extent for the most part. The plants, when they break down, right, they become part of the soils. So, in essence, the plants are sequestering carbon into the soils. Okay. And that's a large mechanism for how carbon gets into the soil. It's not the only mechanism, but it's a particularly important one. Well, when we take fossil fuels out of the soils and then we combust them, we're taking all that carbon that's been sequestered in those soils over hundreds of millions of years in most instances, and we're volatilizing them back into the atmosphere, okay, where they can do harmful things like warm the atmosphere and lead to issues like sea level rise via uh, climate change, right? Um, the problem is that when we look at these emissions of carbon into the atmosphere, a large proportion of them stay in the atmosphere for long periods of time. And that can increase the amount of carbon by as much as 2 to 3 parts per million every year at our current rates. So when you look at the amount of carbon globally that moves into the atmosphere, you'll see these terms efflux and influx. So efflux is this movement out 37, almost 38 pentagrams of carbon per year into the atmosphere. So a pentagram is 1 gigaton of carbon. Okay. Whereas the influx back into these sinks is only a little more than 21 pentagrams per year. So this is a fairly significant deficit that's causing a lot of the problems we have with carbon as a whole in the atmosphere, which is really affecting climate change. And that's largely anthropogenically driven because these cycles, in their natural sense, are meant to be in balance, right? And we've thrown that out of balance, unfortunately, with some significant consequences. But understanding how plants work, we can offset this. And plants, in fact, are a big part of this. They do this, and I've used this term before, ecosystem services, right? Things that living organisms or the ecosystem that they live in do for free, which Mattress Mac has said is a very good price. And I agree. So ecosystem functions benefit us. Actually, they benefit all living organisms. And that's because so for instance, when these plants take in this carbon dioxide, right, that becomes part of their tissue. So as that tissue is returned to the ground, that can in turn move that carbon from the atmosphere back down into the soils. And this can occur from above ground biomass and through below ground biomass like roots. We mentioned in class earlier talking about grasses and how many species in the family Poaceae have very deep root structures and a lot of root mass. Same with seagrasses. When we talked about seagrasses, that root mass directly breaks down in the soils, and it's a very quick process. So that's a good way to get carbon down into the soils. Not to get too off on a tangent here, but that's one of the reasons as we're looking at biofuels as one way to help mitigate or actually rather reduce carbon dioxide in the atmosphere. One of the reasons we're interested in um, uh, using grasses, prairie grasses in particular, as a source of material to make biofuels from is because they're perennial, number one, so we don't have to reseed them every year. Number two, they have these very deep root structures, which helps move a lot of carbon and store it underground. While still providing a source of material for us to use as fuel on the surface. So in any event, ecosystem services are directly, again, tied to these ecosystem functions and really are important if we're looking at sustainability, right? And so if you think about sustainability, the ability of both current and future generations to meet their needs, right? While those future generations, the even further future generations are still able to meet those. It's kind of a badly hacked version of that definition, but ecosystem functions are really a really important part of that because pretty much everything we do as human beings depends on some ecosystem service in order for that cycle to be completed, right? Whether that's the provision of a raw material, whether it's breaking that material down, we need those ecosystem services. So when you look at human economies oftentimes don't factor that in. Um, but that's starting to change again because these ecosystem services are free. They're happening in the background anyway, right? They've been going on for hundreds of millions, even billions of years. So I've talked about blue carbon before, and I might mention it again as we go, because here on the coast, this is a really important ecosystem function that our wetland environments provide. Um, so, carbon dioxide, along with a lot of these other greenhouse gases, have been building, and they're building exponentially. One of the things we're recognizing is that the loss of wetlands has attributed to this because of how much carbon wetlands actually sequester out of the atmosphere back to the soils. So less wetlands equals less sequestration, which means more buildup of carbon. So restoring and protecting these habitats And establishing these plant communities so that they can perform this ecosystem function is to our benefit because that's less plants we need to build, carbon sequestration plants that cost capital to build. They take a footprint, they have to operate. So you have all of the operating costs. They have to be maintained. So you have those costs. So in addition to the capital costs, you've got all of these other costs, and the amount that they do is somewhat limited, right? The amount of carbon they can actually sequester is somewhat limited. Whereas these plants are, to a great extent, if we're not actually harming them with our own actions, are to a great extent self-healing, right? They're constantly doing what they do because that's how they've evolved. So these sequestration rates vary, however, depending on the type of plant you have. Turns out mangroves and salt marsh are two of the most valuable in terms of carbon sequestration along our coasts, right? Seagrass also play an important role. And these all dwarf tropical rainforests, which you hear a lot about and are important. I'm not saying they're not, but they are but they're dwarfed in terms of how much carbon they actually sequester from the environment. So there has been a move to monetize this, right? So monetization occurs across everything. You might be most familiar with it in terms of monetizing videos on YouTube or TikTok. But there has been a lot of effort to monetize carbon because in our human economies, the way we value things is by how much they cost in terms of cash, right? And so um, there has been an effort to monetize carbon in a way to put a price on it so that in a permit system, for instance, you're required to offset the amount of carbon that your whatever your manufacturing process or your actions are putting back out into the environment. You might have seen this. Conferences you can travel to, airlines actually are starting to do more of this, where you can buy a carbon offset along with your ticket, right? That offsets your portion of the carbon that flying that airplane entails. So here in the Texas coastal area, and actually along, this is a global effort, but it's particularly strong in the Gulf Coast, and there's a big movement here in the Texas area to provide carbon markets where you can buy these offsets. And blue carbon, it's called blue carbon because it's in the water. So blue carbon ecosystems are responsible for removing 10 times more carbon dioxide per hectare than, which is about 2.5 acres, by the way, to the atmosphere. I'm sorry, from the atmosphere back into the soils than forests are, right? And they primarily store the carbon in the soils. You don't have that litter layer so much that it can stay in those soils for centuries. A lot of the soils, a lot of the fuels we're burning now have been in the soils for hundreds of thousands of years, right? The problem is that wetlands, when they're drained, so if they're like prairie pothole wetlands that are being converted into housing, subdivisions, or whether they're coastal wetlands that are being degraded through erosion and subsidence, this can take that stored carbon and actually allow it to volatilize back up into the atmosphere. Decomposing plant material actually is a good source of methane, and other volatile organic carbons to the atmosphere. So the objective is to keep them locked up in the soils, right? So this is marketable, as I said, and if you look at just 10 years ago, carbon offsets were about a 300 and almost $400 million market, locking up about 87 million tons of greenhouse gases out of the atmosphere. So that worked out to about $5 per ton of carbon dioxide, metric ton of carbon dioxide. Here in last year, 2023, the averages were about $7 a ton. So that market is going up now. It has fluctuated quite a bit. So there's some years where it's down, some years up, like all markets, right? It's supply and demand. But overall, like markets, we're seeing those values are starting to they're starting to increase. And there's estimates that by 2030, because of the amount of mitigation and counteraction that needs to occur in order to deal with the issues surrounding climate change, Those markets are likely to get stronger. Those carbon offsets are going to be more expensive. So it's estimated to be anywhere from 25 to $30 per ton of CO2 in just about 6 years from now, right? So that's a pretty significant increase. And that makes that market something that businesses are going to want to get involved in. Because the buyers include a wide array of things from our economic system: government, corporate entities, energy, transportation, finance, insurance providers are a big one, right? And we're seeing a lot of movement in here. And again, I know I've said this before, but if you ever have the opportunity to take an environmental economics course, I would strongly encourage you to do it. I think everything that happens in the environment, especially things that are anthropogenic, oftentimes are rooted, in markets. And so understanding a little bit about economics actually can help you be a better environmental scientist or marine biologist. You can find out a lot more information, by the way, if you go to these resources. One thing I want you to bear in mind, though, when you're looking at these numbers is that vehicles are, they're one of the big sources of carbon dioxide to the atmosphere, but they're not the only big one, but they do make a good metric because most of us drive in the United States, right? And we are one of the biggest emitters of carbon to the atmosphere. So passenger vehicles, gasoline powered passenger vehicles, typically are emitting anywhere from 3 to 11 tons of carbon dioxide. So you think about a small economy car or a hybrid might be on the lower end of that. Pickup trucks. Think about how many people that drive pickup trucks, right? Not including yourselves, or your dear professor. So they produce on the higher end of that. Every 15,000 miles, you're looking at 3 to 11 tons. So it can really, there's still a long way to go in terms of being able to adapt that, but blue carbon could certainly be one of those markets. So with that, don't forget about your weekly quiz. It's going to be on this area of biogeochemistry. We'll come back. We're going to talk more about nutrient cycles next class period, and I will look forward to seeing you next time.